Hello and welcome to the second ever episode of Top Notch Science. I'm your host Joe Blazek, and today we're going to be discussing something pretty cool in the life sciences area. There's a group of Swiss researchers over at the EFPL School of Life Sciences that's been working on a technique that allows them to print a construct that resembles human tissue and has the same functionality. Now, in order to do this, they're making use of a three-dimensional printer. And before we go on a little bit further, a little bit more on how they're doing this, we're going to discuss how a three-dimensional printer works. Now, it works pretty much the same way that you would think that a regular printer works, except it differs in one little way. Now, a regular printer basically deposits little pieces of ink or material, as they use in three-dimensional printers, and the materials vary from plastics to metals, to this case, biological matter. And it basically puts a whole bunch of these little dots side by side, and on a regular printer, that's it, it's done then. Now the 3D printer differs because it basically lowers the tray or the paper that it prints on, and then continues to print another layer on top of that, and then another layer, and another layer, and another layer. And by doing this, they eventually create a three-dimensional object. Now what this group has done is create a base for the material to create the tissue. And what's important on this tissue is that when you actually deposit it, it sticks and stays in its original form. Now you want to stay in its original form because if you just simply mix all the ingredients together for a cell and it just goes around like that, the cells themselves don't form properly, and you don't get cells that function like normal cells. Now because of this, you get some very awesome stuff happening. The various tissue elements don't blend together, and you can actually create some three-dimensional structures. Some three-dimensional structures, for example, like channels, which will allow the cells to actually function like they're supposed to, basically transferring fluids in and out of the cell to let nutrients in and let the waste products go out. Now all of this has never really been done before because cells are very complex. They have many different functionalities throughout their lifetimes. They move around, they multiply, proliferate, and they even die. So putting all of this into one sort of material to print is very difficult. But this group has created the basic starting blocks that they need by creating the basis for which they can create all those different channels that the cells will need to function. So now we come to the important part. Why do they want to do all of this in the first place? So, basically, if you can create a sort of little model that very closely resembles human tissue, you can use it to test drugs. Now, not, recreational drugs, but medical drugs for all kinds of diseases, cancers, things like that. Now, currently they're using a lot of animals to do this testing, and you know how messy animals are. They poop, they make noises, it requires a lot of care. Basically by doing this, it not only helps animals from being tested with drugs, but it can be a lot easier on researchers to test very large amount of cell types in a very quick way without having to deal with the whole issue of taking care of all the animals. And again, by having all that functionality, they can test a lot more drugs more cheaply, which means you, the end user, gets much more affordable access to healthcare. And that's always a good thing. So that pretty much wraps up today's episode, and you can probably expect another episode to come on Wednesday.